Good afternoon, everyone, if you are in Australia, and good morning if you're in the UK. Welcome to our webinar series. The CSTFA is a research provider for sustainable tropical fisheries and aquaculture research globally. We provide world-class multidisciplinary solutions-focused research for Australian and international resource managers, both in government and the private sector. We're based at James Cook University in Townsville, in far north Queensland, Australia. I encourage you to check our website to find out more about us or send me an email later on. Today, it is my great pleasure to welcome Dr. Adriana Humanes to speak to us. Adriana is a postdoctoral research associate at Newcastle University. She's currently working on the project Coral Assist, looking at assisted gene flow as a potential tool to be used in coral restoration efforts. Today, she will present a technical workflow for rearing corals from eggs to adults as part of a selective breeding program, using Acropora digitifera as a model species. She will discuss methods for choosing colonies to conduct selective crosses, rearing coral larvae en masse, controlling larval sediment, enhancing early survivorship in ex situ and in situ nurseries, and outplanting the resulting colonies in natural reef substrates. So thanks very much for being here, Adriana, and I'll just hand it over to you.
Thank you very much, Cecilia. Thanks for such a wonderful introduction. And also I wanted to thank each and every one of you for attending this webinar. It is incredibly heartening for me to be invited to this webinar series at JCU, where I conducted my PhD studies. Today, I'm going to tell you a bit about the research we're conducting at the Coral Assist Lab at Newcastle University, where I hold a postdoc research associate position. This project has been funded by the European Research Council. I hope you enjoyed the presentation, and bear in mind that it's 7 in the morning here in the UK, so if something doesn't make sense, it's mostly because I'm asleep at this stage still. So the work that I will be presenting is being conducted by the members of our lab group. The PI of the project is Dr. James Guest, and Professor Alastair Edwards, John Bithell, and Akin Troyman are the co-investigators in our group. We collaborate closely with our local partner, Dr. Jean-Man Golbu at the Palau Coral Reef International Center. In the group, we are 2 postdocs, Dr. Pavel Palmowski and myself, and we have currently 3 PhD students, Evelyn van der Steeg, Liam Lachts, and Elizabeth Beauchamp. We had last year a master's student, Ana Sofia Lorda, and Elios Martinez is our field research assistant. If you want to know more about the research we're conducting, you can visit our webpage and our social media accounts. So first of all, I would like to introduce the subject by talking about why should we care about coral reefs, besides of the fact that they are extremely beautiful ecosystems. So let's see reefs by the numbers. Coral reefs support more than 500 million people worldwide, accounting for 15% of the gross domestic product in more than 20 countries. They're home to a quarter of all marine species, having a representative of 28 out of 35 animal phyla. This is why they are so incredibly beautiful, because they sustain a diverse community of organisms, making them an ideal place for marine ecologists like me to study them. In addition, since the 1950s, around 20,000 marine natural compounds have been described, and many of them have been used to treat diseases like heart disease, cancer, asthma, and arthritis. In this map, you can see geographically where are the countries located where human populations depend from coral reefs and get benefits directly for food or coastal protection from them. As you can see, it is the Middle East, the Southeast Asia, and the Caribbean, the places where more people depend on this ecosystem. The great majority of these places are low and middle income countries, where access to funds for technology and to manage these ecosystems is a real limitation. Sadly, as you might be aware from the news, coral reefs are facing serious threats. A recent study from the National Oceanic and Atmospheric Administration published in 2018 a report saying that approximately 75% of the world's coral reefs are currently threatened by local and global pressures. And as a consequence, corals is the group moving towards increased extinction risk most rapidly, according to the reports by the International Union for Conservation of Nature. So coral reefs are affected by local pressures like overfishing, bycatch, and destructive fishing, ocean pollution and coastal development. And currently the biggest threat to coral reefs that are facing is climate change. As you might already know, the production of carbon dioxide from anthropogenic activities has occasioned the increase in global temperature by almost 1 degree. As you can see from this graph, where the carbon dioxide is represented in the yellow line, there's a significant correlation between the increase of CO2 in the atmosphere and the global temperature here represented in red. Nonetheless, we are really lucky to live in a planet with such a huge surface of water, because water is a better conductor of heat than air. And as a consequence, oceans have absorbed 93% of the heat emissions generated by human activities and 25% of the carbon dioxide. But this has resulted in an increase in the seawater temperature during the last 2 centuries. In this graph, we can see the anomalies in temperature from the average between 1961 and 1990, represented with the red and the blue bars. The sea surface temperature anomalies in tropical areas, shown here in the red line, are becoming more frequent, last for longer periods, and are reaching higher values through time. As a result, the average annual degree heating index, which is a measure that we researchers use to estimate the accumulated heat in the seawaters, has been increasing over the years in the tropics, as you can see from this graph. And in here, I've added red arrows to indicate the years when massive coral bleaching events have been reported and studied in detail by the scientific community and have affected hundreds of reefs around the world. As you might be aware, only during the last massive bleaching event, the Great Barrier Reef has lost already 50% of its coral cover. And it's been estimated that during the last 30 years, coral cover worldwide has been lost around 20%. The Intergovernmental Panel of Climate Change, which is a global consortium of world top climate scientists, published in 2018 a warning that unless that we limit our future warmings to 1.5 degrees, there is going to be a very bad consequences for coral reefs. And even if we can limit to 1.5 degrees, it is highly likely that a large areas of reef are going to be experiencing regular bleaching events, threatening between 70 to 90% of the reefs by 2050. So without drastic interventions, we risk losing most of the reefs. Global mean sea surface temperature is projected to reach 1.5 degrees above pre-industrial periods, about the pre-industrial levels between 2030 and 2050, depending on the model and assumptions regarding projected changes of the atmospheric greenhouse gas levels and the climate sensitivity of the model. What this means for coral reefs is that they will have between 20 to 30 years to adapt to the projected temperature increase. For some coral species and reefs, this might not be enough time for natural selection to act and select for the most tolerant individuals. So with this background, how can we help reefs to face climate change, considering that they might not have enough time to adapt to the predicted scenarios? In order to conserve coral reefs as we know them today, we need a portfolio of different strategies to tackle simultaneously the causes of the decline and to deal with the symptoms that climate change produces on coral reefs. The main action is a reduction in greenhouse gas emissions. And the ways of achieving reductions in greenhouse gas emissions needs to be led by each nation government and also by each one of us as individuals. Another action that will help conserve and protect coral reefs is the reinforcement of local and regional management programs of marine areas to maintain and reinforce connectivity between populations and metapopulations and to develop plans for risk mitigation. Active restoration efforts of degraded reefs, it's another of the actions to restore these affected areas or to prepare them for future heat waves that is part of this portfolio of options to conserve coral reefs. In this context, marine ecologists have been focused on doing research to better understand the different roles that each one of the members of the coral holobiont play. The animal host, the algae, and the microbiome, and what makes a resistant coral holobiont. Is it a certain species of a zooxanthella or a particular microbiome? Or is it that the animal host has a specific genetic information that makes them resistant? And being the case of any of these being important, how can we use this information to help reefs face climate change? Assisted evolution has been proposed as the framework to understand heat tolerance and considers 4 different approaches. The first one focuses on inducing acclimatization by exposing individuals to the stress condition, improving their performance by making them more tolerant to high temperatures. A second idea is to modify the coral microbiome, since it has been shown that certain microbiomes can be associated to heat resistance. A third approach is to perform selective breeding process, where corals that are known to be resistant to temperature stress are selected to produce new individuals that are going to be used in restorative activities, the same way that we humans have done to improve the production of plants and animals for human consumption over centuries. The fourth option is to provide corals with Symbiodinium algae that are known to be more resistant to high temperatures. The final goal of these 4 approaches is to produce stress-tolerant individuals that are pre-adapted to future stress conditions and that can be introduced to natural populations. In Coral Assist, we're focusing on selective breeding to investigate several hypotheses related to heat tolerance. During a bleaching event, it has been observed frequently that colonies can show differences in their thermal tolerance in some individuals. Some individuals are able to survive the stress event better than others. In this picture, we can see 2 colonies from the same species, one next to each other, and they've been exposed to the same stress condition during the mass bleaching event of 2010 in the Caribbean. But in here we can see that one of them is completely bleached while the other one looks healthy. This difference in heat tolerance can be used for perform selective breeding process within individuals from one single population. It is important to keep in mind that we are not introducing new genotypes into the population of study or performing any gene editing. The main goal of this approach when we work with individuals from the same population is to increase the proportion of colonies that naturally occur in the population with a phenotype that is resistant to heat stress. We assume that this approach will be beneficial for those populations where the proportion of reproductive adult colonies is reduced by the effects of local and global stressors, compromising the reproductive success of the population and the potential effect of natural selection to occur to actually select for more tolerant individuals. So using this framework, we are focusing on the following questions. First, we want to know if heat resistance in adults can be passed to the offspring. Second, we are interested in getting more insights into the causes of variation in heat tolerance in corals. Third, we want to know if there are any costs associated to heat resistance. For instance, are there any trade-offs between heat resistance and survivorship or fecundity or growth? And fourth, we're interested to know or explore if heat tolerance can be used in restoration efforts. I'm going to focus the rest of the presentations in experiments we have done to answer the fourth question. And I will go through the main challenges that we have faced and the results we have obtained so far, focusing always on the need to develop methodologies that can be used by local managers with minimum access to technology. So our project is taking place in Palau, which is a country located in the Pacific Ocean, consisting of 360 islands in an area of 466 square kilometers. We are working in reefs in Palau that are either this type of reef in protected lagoons or exposed offshore reefs. In there, we're working with 3 species that have distinctive life histories and growth forms: Echinopora lamellosa, Goniastra retiformis, and Acropora digitifera. I'm going to focus the rest of the presentations on the results we have from Acropora digitifera, a branching coral that is common in shallow reefs area in the Pacific. So in order to identify the colonies that have distinctive heat tolerance without the incidence of a bleaching event, we have conducted heat stress tank experiments. For this, we have tagged around 160 colonies, adult colonies in the reef, and we have taken 6 branches, small branches of each one of these colony, and brought them to the research station, leaving the donor colony on the reef. To perform the heat stress, we have exposed these fragments to a temperature stress profile similar to the ones reported during bleaching events and this has been done under tank conditions for a time frame of 5 weeks. And to do this, we have used just basic aquarium equipment, heaters, lights, and plastic containers as tanks. During the course of the experiment, we have assessed the health status of each fragment on a daily basis, reporting if they are looking healthy, if they are starting to bleach, if they show any signs of partial mortality, or if they are dead. And with this information, we have estimated a bleaching and mortality index. As you can see from this picture where we have fragments from 2 colonies, on the top we have a colony where the fragments have gone through a bleaching process throughout the time of the experiment, while the colony in the bottom is not showing any signs of change in its health status. So this is the way that we analyze the results of these experiments with a clustering graph. And in here, you can see how the bleaching and mortality index varies across time for each one of these 104 colonies tested in this experiment. So each one of these rows represents a colony, and each column represents a time point in the experiment, in the heat stress exposure. The blue indicates values that are corresponded to an index of being relatively healthy at that time point in relation to the status of the fragments from the other colonies in that same time point. So for instance, in this, in red is indicating that the relatively health status of the fragments of that colony is relatively unhealthy, meaning that it's either showing signs of bleaching, partial mortality, or is dead. So for instance, in this time point, this colony was the one performing the worst in all the fragments exposed from that colony in comparison to the rest of the colonies during that time point. And at the end of the experiment, we can see which are the colonies that are doing considerably good during the heat stress against the ones that are doing considerably bad. And the good ones are the ones that we are going to consider to be relatively high in heat tolerance because they haven't shown any signs of change in their health, while the ones that are showing changes in health are going to be considered the ones with relatively low heat tolerance. So one important aspect to consider when using this methodology for restorative activities is that previous knowledge of the environmental conditions in the reef of study and the population dynamics will be extremely beneficial. For example, we have named the reef where we work in, we are studying Acropora digitifera in Palau. We have called this site the washing machine because of the strong wave action in the reef crest where the tag colonies are located. As a result of this harsh environment, we have lost approximately 36% of the colonies that we have tagged throughout 1 year. And this is because either they have lost their tag and we couldn't relocate them, they have shown signs of partial mortality and therefore we don't want to use them anymore for our experiments, or they have gone through death. So special considerations need to be taken when planning using this type of methodology. If we want to have a minimum of 6 parental colonies for doing the selective breeding process, which is the minimum amount of colonies to have to capture the most common alleles in the population. Another factor that needs to be considered when planning these experiments is that we need to prepare the substrates that are going to be offered to the resulting larvae. So conditioning these substrates is critical, and in the literature it has been established that a minimum of 6 weeks before the spawning is needed to secure the development of a biofilm that results attractive to the coral larvae. This can be done either in the reef where we're working in areas where CCA or crustose coralline algae are abundant, which are known to induce coral settlement, or it can also be done under aquarium conditions. So when the corals are predicted to spawn, what we have done is we have brought the colonies to the lab, the ones that we have selected as with distinctive heat tolerance, the highest and low tolerant individuals. And we have collected the gametes when spawning occur to perform the desired process. The collection of the gametes can also be done without removing the colonies from the reef by placing collecting nets on top of the colonies and collecting their gametes. But due to logistical conditions, since I mentioned that this is an extremely harsh environment, the washing machine, we were not able to place the nets on top of our tag colonies. So we had to bring them to the lab. So once the gametes are obtained, we have produced the selected crosses that we were interested into studying. And this gives the opportunity because in this case, Acropora digitifera is a hermaphroditic species. You have an individual producing both gametes at the same time. So it's possible to do crosses with the female gamete is present or the male gamete is present in the cross to test heritability questions regarding the influence of maternal or paternal effects into the heat tolerance. So for the gamete fertilization and larvae rearing, we have used 3 different setups that go from very simple static pockets without any flow through, which is what has been used since the beginning of research in coral spawning back in the 80s. And this is a really inexpensive and affordable way of doing coral spawning that is available in low-income countries. and does not require much technological expertise. And we have also used more sophisticated designs, and like the conical hatching jars that is shown here in the picture, that has flow through and that allows us to control a little bit better the conditions in the cultures of the larvae. We have also started doing trials, testing different settlement densities for obtaining appropriate number of juvenile colonies per substrate of per unit of substrate that we want to use to outplant to the reef, and also to avoid the formation of chimeras and density dependence mortality. And this has also been done in a really simplest way. So nowadays, there's a lot of scientific knowledge related to gamete collection, fertilization, larvae development, embryo development, rearing of these embryos. At this stage, researchers are able to get really high fertilization success, close to 90%, and similarly, good larvae survivorship under laboratory conditions. However, there are still many unknowns related to the settlement cues and drivers of early survivorship, both in captivity and in the reef. And these two processes are the major bottlenecks that are driving coral population dynamics. So ideally, what we would like is to produce coral juveniles that can be successfully transplanted to the reef as soon as possible, to decrease the effort and time and the cost needed to maintain them in captivity conditions. However, in the meantime, while we are testing different approaches to overcome this bottleneck, we have maintained juveniles in ex situ nurseries, like the one shown here in the picture, where we are providing a good intensity of light, and with the presence of herbivores like rabbit fishes and snails, and also urchins that maintain the algae community at really low levels in this tank and promote the clean areas and spaces for the growth of the coral juveniles. So we have tested different outplanted ages of these coral juveniles into the reef. By using juveniles from the same age and maintaining them under tank conditions for different time periods. In this graph, you can see the results of outplanting individuals after spending 5 months in the tanks or 11 months in the tanks. And as you can see, there's a significant effect on the juvenile survivorship when they're outplanted really early to compare when they're outplanted at a much later phase. So the individuals that are outplanted later, they have increased their survivorship almost by from 6% to 40%, which is a great increase. And the good news in here is that in both cases, there were no significant differences between the two treatments that we have, the different type of crosses these individuals came from, either from parental colonies with high thermal tolerance or low thermal tolerance. In both cases, there was no difference in their survivorship among the treatments, suggesting a lack of a trade-off at this stage between heat resistant to temperature, heat resistant and survivorship. So another interesting result from this experiment was the sizes that these juveniles have grown to. These pictures show the final size at this stage. So when they've been, when they are 24 months old, and this is the last survey that we have done this last June. And as you can see, the ones that were outplanted really early into the reef have not been able to grow that much, and they haven't even started branching. While the ones that were outplanted when they were 11 months, they're already showing a great increase in their growth, and the great majority of them are showing more than 5 branches already. So processes of herbivore and competition with other invertebrates have been reported in the literature to determine these differences in early survivorship and growth in coral juveniles. Moreover, what is really interesting as well is that if we look at another set of colonies from the same cohort, from the same batch of individuals that were produced and were used in the outplanting to the reef, but these ones were kept for 11 months under tank conditions, and then they were transplanted to an in situ nursery that was caged. In here you can see the nursery without the mesh on the top, but it was caged to prevent herbivory of big fish. And as you can see, when you compare the sizes and the growth of these colonies, they are already looking more similar to an adult colony in the reef than the ones that were outplanted directly to the reef. So we can make the analogy of the tank conditions being similar to a 5-star resort where juveniles get good light and food conditions and minimum stress levels. While the in situ nursery might be more similar to a budget hotel where light and food are good, but there's already some stress conditions that it's driving the growth of these individuals. And the individuals that were transplanted directly to the reef is equal to thinking that they were like going camping into the wild where stress conditions are extremely high and they need to cope with a lot of pressures from different factors. So to address the question of how could we develop methods that could be used to maximize the survivorship and the growth of outplants, if we want to perform these outplants at that early stage or early age of the coral, and to overcome these bottlenecks in a cost-effective manner, we're working together with SECOR to test different types of substrates that can confer an increase in the survivorship and the growth to the individual colonies. In addition, one of our PhD students, Evelyn van der Stek, is focusing part of her research on testing substrates that can offer protection from herbivory and competition with other invertebrates during the first months of life of a coral, and also promote and also be a method that can be used at relatively large spatial scales. So in here, I'm showing you the results that we have obtained with 1-year-old juveniles that were exposed to a heat stress event. From an evolutionary perspective, the goal of these efforts in using selective breeding is to establish a self-sustaining and sexually reproducing coral population that has a sufficient genetic and phenotypic variation to adapt to a changing environment, and that is sexually reproducing. So one encouraging result that we have obtained so far has been the response to heat stress of individuals 1-year-old that were produced by selective breeding process. In this graph, you can see the colonies highlighted here in blue that come from crosses from parental colonies with high heat tolerance, and they are the ones that are performing better in the exposure to the heat stress in comparison to the individuals that were produced by crosses from low heat tolerant parents. So there's some evidence in here showing us that heat tolerance has a heritable component. We are aiming to conduct more studies, perform more heat stress experiments on individuals from these crosses when they are adult reproductive colonies, whenever we're able to go back to Palau if Corona let us. So another question that we're trying to answer is where should we outplant the colonies that are produced by selective breeding, and how many of them should we outplant in order to have an ecological impact in the population. And for this, we are collaborating with Dr. Renata Ferrari from AIMS and Dr. William Figueira, Dr. Bridget Sommer, and Daniel Pigas from Sydney University. They're using 3D modeling by constructing orthomosaic maps from the reef where we're working, and trying to establish, characterize the coral community in the reef and the position of each colony in the reef to correlate the survivorship of individuals to the position where they are. In this way, we will be able to answer, for instance, what are the good places to outplant colonies? And when we could use this information for doing population models to project how many individuals need to be introduced to maintain the population at good levels. So I want to finish with this final slide representing the workflow that is needed to implement a technique for ecological restoration. So all the results that I have presented so far are just proof of concepts. It's just an example of how restoration ecology works. Restoration ecology is the science that supports the practice of ecological restoration. So in here, we're not doing any ecological restoration. We are just proving concepts that can be used in ecological restoration. And it's important to highlight that only a few projects, a very few restoration projects, have been successful in attaining any good results at large geographical scales through time. So to develop an ecological restoration project, which is the process of assisting the recovery of an ecosystem that has been degraded, damaged, or destroyed, there are mainly 4 major components involved in its design. So first, using the knowledge from restoration ecology and with the appropriate financial and institutional support, restoration goals need to be defined. And this needs to be done together with the stakeholders and the local community. So firstly, we need to know what is the goal of this restoration project and define how these goals are going to be measured. Then to actually proceed with the ecological restoration project or restorative activity, which could be, for instance, rehabilitation of a reef area. The project needs to, there needs to be a plan on how it's going to be executed. And one important thing to consider in here is that the processes that occur in coral reefs are usually processes that might take a few years, at least between 5 to 10 years, to be able to see an impact in the community that we are working with, in the coral reef community. So these projects need to be planned appropriately in a way that enough capacity building is provided to have the people that are going to be executing the plan. And usually these are practitioners from the local area. Then they need to implement the activities related to the restoration project. And after they have implemented the activities, there needs to be some monitor and evaluation of these activities and the defined indexes that were going to be used when the restoration goal was defined to measure the success of the activity need to be used to evaluate the implementation of this project. And then there needs to be a phase of readjusting the execution of the plan, considering the results that have been obtained during the first years of the project. So with that, I wanted to finish acknowledging all our collaborators and supporting institutions. Coral Assist is only possible thanks to the work of our lab members and the great group of scientists and aquarists that work together with us. Sulang, which is how we say thank you in Palawan.
Thanks very much, Adriana. Now we're open for Q&A, so if anyone would like to make a question, you can just type it on the Q&A panel. And while we wait for people to type their questions, I wanted to ask you something. So you used Acropora digitifera, some other species, I assume because it's a fast-growing coral?
It's a fast-growing coral, but also it's really abundant in the Pacific. But not only that, one of the main questions that we have is what are the characteristics that determine heat tolerance? And for this, the second postdoc in the project, he's a specialist on proteomics. So we're looking at the protein expression of these individuals from different groups. And in order to do proteomic analysis, you need the genome. So Acropora digitifera is one of the Acroporas, like we wanted to work with at least one Acropora. And Acropora is one that has the genome available, is high abundant in the reefs in the Pacific. And the other thing is that we spend tons of times in the reef maintaining these transects. So it needed to be a species that is abundant in shallow areas that wouldn't limit our time in diving, and also the cost associated to that. So yeah,
so
that was basically the reason for choosing Acropora, but we're doing the same, like we're using the same approach with Goniastria and Echinopora. It's been harder to work with the other two species. The other thing is that we knew when Acropora digitifera would spawn in Palau, which was really important to develop any of this work. And for instance, for Goniastria retiformis and Echinopora, we were not that sure when they were spawning. So actually we only, so Coral Assist started in 2017, and only last year we were able to collect gametes from Echinopora lamellosa.
And for longer lived species, if you have a longer lived species, do you think, how feasible do you think this is? If you have to wait for the corals to be juveniles and to develop for a year for Acropora, for a species that has slower growth, how feasible do you see this?
Yeah, so for instance, massive species, they are much longer, they have a much longer lifespan. But we were able to reproduce Goniastria, and we have juveniles of Goniastria, and we have outplanted them into the reef now. Evelina, the PhD student, is working on that species specifically. And there are reports, for instance, from the Philippines, from the collaborators from James Guest, that they have found massive species to be reproductive already after 4 years. So it can be that they reach adult stages much faster than what we have thought. It's just that they are going to be really small in size still. So doing these approaches with massive species, it's going to take a much longer time for you to be successful to actually use it for restorative efforts. However, we also know that, for instance, massive species are more resistant in general to heat tolerance. So maybe there's this trade-off where do you want to invest the money? In something that you know that it's going to resist better to high temperatures, or something that it's more fragile to high temperatures. But on the other side, you also want, for instance, you could establish this type of programs to target those species that you know that are going more likely to be disappearing because of the heat stress. So Acropora species are more ideal in that sense. you will expect natural selection to act on mass species.
Yeah. We have one question from the audience, from Ricardo. I was wondering if in your project you also use colonies already resistant to high temperature and maybe pH. I was thinking to Nico Bay.
Can you repeat if you're using resistant?
I was wondering if in your project you also use colonies already resistant to high temperature and maybe pH.
So no, we're not working with he's referring to the colonies from Nico Bay. Nico Bay is an area in Palau that is exposed to high pH, and we didn't work with that population exactly because of that, because we didn't want to have two factors that would confound our results. So we're only using heat tolerance at this stage. And the colonies that we're using colonies that we know that are high, so from the same population, but that have distinctive tolerance to to temperature.
We have another question from Julia. Do you already have guesses what makes corals more heat resistant? For example, do you think that they have certain physiological advantages?
Yeah, there's been, the other postdoc, Pavel, he has done some proteomic analysis already from the parents and the larvae. And in the parents, we haven't seen any, so in the first analysis of the results, which is not in depth yet, but we haven't seen any differences between adult colonies with high and low tolerance when they're adults. But at the larval stage, they're already showing like really different patterns in terms of the proteins that they're expressing. We still don't know if this is related to heat tolerance or not. We need to conduct more studies, but they seem to be clustering in different groups. So that's already showing some explanations on differences between the two groups.
And genetically, if you just look at the DNA structure, there's no structure or there's no major differences between the heat resistant and the non-resistant?
We're still waiting for the results from the genetics, and hopefully Corona lets us get some results soon, because it's really interesting to know what is driving these differences. So we are in the process of getting those results.
We have another question from Julia. Is this different protein expression based on the host or the zooxanthellae?
So the difference was on the host, because the difference that we were looking at larvae that don't have any zooxanthella at that stage. So it's mostly, it's just the host response. Yeah, great question. It's like, because there are the three components of the holobiont. So by analyzing one good thing is that we can analyze the response in the proteomics of the gametes individually, or of the embryos or the larvae, and that will give us some information about what the host is doing without the zooxanthella.
Julia is saying perfect, thank you. And I wanted to ask you as well, for the 5-week experiment, when you determined which ones were heat tolerant and which ones were not, do you collect all those colonies from the same area?
Yeah, they're like, we're working all the time with the same population. It's a reef, and we're collecting everything. Like, we have 12 transects, and the separation between transects goes between 5 to 10 meters in between transects. So they're really close, and we're mapping the whole area to know where each one of these colonies is. One of the hypotheses that we have is maybe the heat tolerance is like in groups in the reef, like maybe some areas are the ones supporting like individuals that have higher tolerance, but they are all located more or less at the same depth. The depth is between, let's say, one meter and 3 meters. So it's not that huge difference between colonies, but we are in the process with the 3D modeling team to analyze exactly the location and relate the heat tolerance to the depth and the location in the reef to see if we can see any patterns in there. But we have conducted these studies, like we have done heat stress experiments already with the 3 species, and exposing, I think, around 300 colonies, more or less. And in each one of the populations we have worked, because we were working with one population of Acropora, one population of Goniastria. And for Echinopora, we are working with 3 separate populations. And for each one of them, we always find around a 10% of the individuals to be in the extremes of the distribution of the heat tolerance. So either high or low.
We have a question from Liam. Hi, Adriana, great talk. What could be the pros and cons of doing selective breeding from within one population, as you have suggested, or doing more assisted gene flow options, where corals are moved between locations?
So I think one of the problems might be, for instance, if we're working with cases like in the Caribbean with Acropora palmata or Acropora cervicornis, which is a population that has been really decimated, and there are really low numbers of individual genes, or there's a huge depression in the gene pool. So if you work with only one population in that case, you might be limiting the diversity, the genetic diversity in that population. However, if you're working with populations that are abundant and are not affected by their distribution, there shouldn't be any problem related to genetic drift or genetic problems associated to selecting the individuals to do the crosses. Another problem that I could think, well, another problem that is more like methodological problem is like, imagine you establish you tag your colonies and you have done all your experiments to find out which are the colonies with the characteristics that you want to use for the selective breeding program, and something happens in that reef and everything dies, for instance, a cyclone, then you don't have any more material to work with. Well, if you are not limited to just working with one population, but one area, for instance, like the examples of the work that has been done by AIMS, where they do assisted migration, where they select individuals from populations that are known to be exposed to higher temperatures. So you are not limited to one certain reef, but you could select colonies from a certain area. You wouldn't have that problem of limitation of the availability of the colonies.
And has your group done genetic assessment of the corals along at a higher depth?
So in the reef where we are working, Acropora digitifera doesn't occur at higher depths. It stops around 3.5 or 4 meters. After that, we have other Acropora species. So no, and we haven't seen Acropora at deeper sites in Palau in big numbers to be able to actually like have this type of study. However, for instance, with the Echinopora, we do have like the reefs where we're working. It's in those inner lagoons, and the depth for those individuals, they go up to 9 meters depth.
And there's no, is there gene flow between the colonies at shallow and deeper areas?
For Echinopora?
Yes.
Yeah, so we still don't have any information related to gene flow for these colonies, like, yeah, getting results. We're waiting for getting results, but it's also like really challenging to work with these three species at the same time. It's a lot of work involved and samples to be taken and all the logistic of posting samples from Palau to the UK, not easy.
I can imagine. And now, especially with COVID-19, that's an extra level of complexity.
Yes.
Well, thank you very much, Adriana. That was a really great webinar and great work that you guys are doing at the Coral Assist Lab. Thanks very much for joining us, everyone.